There's no scientist that I know who hasn't asked him or herself, well, what do we do that might be helpful? So what's important to appreciate is that in all viral infections, there's a tremendous amount of interaction between the virus and the host. The virus has to hijack all sorts of host processes in order to infect a person and make them sick. And the hijacking of all of these host processes actually represent opportunities for intervention. If the virus needs to latch onto a protein on the surface of a cell to get in, well, if you interfere with that latching process, then you can protect against infection. Right now, a lot of the companies are focused on two approaches. One is, well, just to go after the virus directly. Remdesivir is an example of that. Another approach is to go after the proteins directly that are human proteins that the virus needs. We're trying to add a third dimension to these development efforts, where instead of going after the virus directly, instead of going directly after the host proteins, we're going after the expression of the genes that encode the host proteins. So we're just trying to make less of the protein there in the first place. We used a strategy that is often referred to as repurposing. So the idea here is what we'd like to do is find something that's already available that could be used immediately. So when we ran these analyses, we had a really clear set of results that really suggested the best possible direction from the data that we had so far, therapeutically. And those results suggested that the best opportunity was to try to reduce the activity of this one particular host protein that the virus needs, called TMPRSS2. So this is a protein that is on the surface of cells, and what this protein does is processes the viral proteins in a very specific way to allow the virus to get close enough to the host cell to actually have the host cell infected. And if this TMPRSS2 human protein isn't there doing its job, then the virus can't infect. And so when we ran these analyses, we found that there are available treatments that clearly, very strongly downregulate this particular critical host protein. And of all the things that we were able to assess, the strongest, most consistent signal that we found, the strongest things we found turned out to be estrogen and anti-androgen treatment. And that strongly suggested a therapeutic strategy that would use a hormone-based treatment to try to push down the expression of this key protein that the virus needs in order to have people have a less severe disease course because they don't have as much activity of this protein that the virus needs. One thing that I have been struck by, really struck, is just how much of the global scientific community has marshaled its attention to work on this problem. I think that it is unprecedented in all of human history to have as abrupt a change in global scientific attention as we've had in this very short period of time. We are absolutely going to have treatments that help. I think that we, like I think almost all other research groups, we wanted to try to help wherever we had anything to contribute. So we already had a framework set up for trying to find ways to regulate the expression of genes, so we thought we could deploy that. There's no scientist that I know who hasn't asked him or herself, well, what do we do that might be helpful? And I think one should only try to work where one can really make a contribution. So there's no reason for me, for example, to start doing virology. I've got no expertise. I don't know how to do it. I can't help. But I think labs where they actually have relevant expertise, in general, they've tried to deploy it, and that's what we've done.